Great, so it's really a pleasure to be here today and be part of this panel and speak about some of the I think, forward-looking things we're trying to do with 3D bioprinting. You heard a lot of obviously fantastic examples of how 3D printing in some form has been around for 30 years and is really making fundamental changes in the way parts are made, parts are designed, increasing performance. And I want to just show you, moving forward, how that's also going to apply to the way we think about repairing the human body and treating disease, and just some of the potential of that technology with some examples of what we're just really starting to do in my lab at Carnegie Mellon University. If you look on the monitors, this is a picture of clocks from Salvador Dali. All right, so these are clocks that have kind of deformed or flexible. I just want you to think about for a second, what if you had all the pieces to that clock, right, but they were flexible, right? How do you build that clock from floppy pieces, right? It's going to be kind of tough, okay? That's actually the challenge for 3D bioprinting living tissue, right? We have bones inside of us that make us rigid, and you don't realize it, but you're contracting your muscles right now to provide stability. But if you just relax in your chair, right, you're going to flop over. And this is the challenge with 3D printing soft materials. How do you support them during a layer-by-layer process where you're trying to put one layer on top, right? So if you're doing layer by layer, and you put one layer down, and it can deform, how do you accurately put another layer on top? It's almost impossible. Well, what we've recently developed in my lab is a way to do that. We call it free-form reversible embedding of suspended hydrogel, or FRESH. So these are fresh prints. Okay, so depending on the audience or the generation, sometimes that hits and sometimes it doesn't. But regardless, what we're able to do is we 3D print a gel inside of another gel. Think about hair gel that has bubbles inside, right, on the shelf at the supermarket. Those bubbles will stay suspended in that hair gel forever because they're not applying a force, they're just sitting there. But if you apply a force to the bottle, you can squeeze that hair gel and make it flow out. So that's actually the material in that Petri dish, and we drop a syringe inside of it, a needle from the 3D printer, and we can print a second gel inside of that first gel. And the cool part is the gel we're printing inside of is made out of gelatin, which is thermoreversible, which is just a fancy way of saying at room temperature it's a solid, and you raise it to body temp, it's a liquid. So if this plays, oh, excellent. So we're printing CMU. That's a gel that we added some black food coloring to so you can visualize it. Otherwise, it would be a clear gel in a clear gel, and that's a boring video. But here we're printing CMU. We do the whole printing at room temperature. This is on a modified MakerBot system, so again, leveraging these open source based technologies. And then when we're done, we raise the platform that this Petri dish is sitting on. We raise the temperature of that, and it melts the gelatin, and it releases what we print. So what can we start to think about doing with that now that we have a process to print soft materials? And we just published this about 4 months ago, so this is very new, and I'm going to show you some of the forward-looking stuff we're trying to attempt. So this is an embryonic heart. So this is a cross-section. Those are ventricles. They're filled with very complex tissue. We're trying to understand how to repair the human heart because heart disease is the #1 cause of death in the world. Very many forms of it. And we've really been trying to work on recreating heart formation using different kinds of advanced stem cell technologies where the US also leads, and trying to integrate these into the manufacturing process. And this is one example of our 3D printed embryonic heart scaffold, and hopefully you can kind of appreciate on the video that we're able to recreate some of the really complex internal architecture of that system. And that one's made out of a polymer called alginate, but we can make that with a wide range of materials. And this really, for the first time, allows us to merge fields that were never merged before. I think that's a kind of a really cool aspect of additive manufacturing. We're taking developmental biology, right, and manufacturing, and putting them together to create new ways to build tissue to treat disease. I mean, these are some of the really, I think, cool things that come out of this technology. This is another example. This is of a blood vessel. So everyone's heard of a heart attack, right? That's basically where a blood vessel that feeds blood to the heart becomes blocked, and then a part of the heart becomes damaged. And you typically need something called a coronary artery bypass graft, right? Which is basically they take a vein out of your leg and they use it to rewire the heart to create a bypass around the blockage. What we can do now is basically that artery is shown kind of on the outside of the heart. Here's a highlight. We can now take MRI data, right? So we do an MRI scan of the patient, okay? Create a 3D model. So now you see this branched network of vessels. And now we can essentially feed that directly into the 3D printer like any CAD model, and now 3D print that out of basically the biological polymer and use that now as a scaffold to regrow the blood vessels. So this is some of what's possible. And this should just be a video showing perfusion through one of these 3D printed blood vessels based off of the human MRI data of this coronary artery tree. So this is far from actually being, I guess, ready to go into the OR, but these are the kinds of new technologies that are enabled, I think, by these platforms. This is another cool example for disease modeling. So what you actually see there on the left is a 3D printed mammary duct network in the Petri dish. This is to study something called ductal carcinoma in situ, or DCIS. This is breast cancer, okay? So of all the women who are diagnosed with this DCIS, 20% will become metastatic disease. 80% will never become metastatic, which means you'll never die from it. But almost everyone gets mastectomy, all right? So, but only 20% actually proceed to invasion. It's because we don't understand why it happens. We're building next generation models of human tissue in the dish to study fundamental disease processes, right? Which will hopefully change how we basically diagnose disease, how we assist patients in planning their medical care with their physician, and ultimately as a platform to develop new drugs to hopefully prevent invasion, even for those women where invasion is a higher probability. Pretty cool stuff. This next example is we took an MRI image of the human brain. We shrunk it down to about 1 inch in size. We 3D printed it. This is just a model of a human brain. This one can't think, or else I'd be using it on myself. Get a little upgrade. But we're dripping black food coloring on top of that to show kind of the complex surface folds of the brain we can recreate. So this is very far from building a brain, but what we can do now with this, which is equally, I think, interesting and maybe impactful, is to model the actual shape and mechanics of a human brain towards better understanding traumatic brain injury, so that we can take what used to be done in computational models and now build a brain mimic and apply similar loads, and we can start to now understand how really complex forces can propagate through brain tissue and ultimately lead to dysfunction. Again, another kind of application of this technology, not for maybe building a part to replace a damaged blood vessel, but just really for this advanced disease modeling in the Petri dish. So those are just a few kind of, I think, forward-looking examples I had, and I just wanted to highlight that we have a really talented group of students that we are, training in the next generation of additive technologies applied to bio and metal and so forth. Andrew Hudson and Andrew Lee are here, and they'll have a table with our bioprinter. We've actually kind of speaking to this democratization, we built all of this on open source platforms, so we've actually released this now back to the open source community so that other research labs can actually build their own bioprinter using this technology for under $1,000. And the interesting thing there is that my lab at Carnegie Mellon can only do a small piece of kind of the future applications, right? And by spreading the technology through this open source maker kind of vision, hopefully this just continues to grow and blossom. So, thank you.